So, hello, welcome to A Moment of Science, please. I'm your today's host, Mohammed.
And I'm your second host, Aghia.
Today we have Dr. Edgar on our podcast, who's a professor in the Temerity Faculty of Medicine within the Department of Pharmacology and Toxicology. Welcome, Dr. Edgar.
Hi, everybody, and thank you very much for having me on the podcast. It's really exciting to speak about science with budding scientists like yourselves. So I'm really excited about today.
Yeah, thank you. So if you could please tell our audience a little about your background, education, and what really interested you about immunopharmacology.
Right. So as Mohammed said, I'm a professor in the Department of Pharmacology and Toxicology here at U of T. I'm actually an assistant professor, and I just started my lab about 9 months ago. So I'm very new. Before starting my independent research group at U of T, I was a postdoctoral fellow funded by NSERC at the Scripps Research Institute in La Jolla, California, working with Professor James Paulson, focused mainly on learning about the multifaceted roles of carbohydrates in immune responses. Before my time in California, I was actually a PhD student in the Department of Chemistry here at the University of Toronto. I was working with Professor Mark Nitz, synthesizing some rather exotic molecules that contained unusual elements. And we use those molecules as probes to learn about how tumor cells actually, I'll use the term breathe within solid tumors, and how much oxygen they might have access to at a given time point. So really combining hardcore synthetic organic chemistry with some in vivo tumor biology. And before that, I was an undergraduate at the University of Toronto, where I did, surprise, surprise, a double major in chemistry and pharmacology. So I've had an admittedly extremely linear and traditional academic career path. And I recognize that I'm very fortunate to have had the opportunity to have such a linear and, I would say, in many ways, smooth pathway to where I am now. And I'm very grateful for that. And it's really great to be back home.
Wow, that's great. Thank you so much for sharing that. I think our audience, who are mostly U of T students like us, would appreciate, seeing that there are so many opportunities out there. And, with your permission, I want to now go a little bit more in depth of your research and research interest. And so we know that obviously as researchers, you probably have multiple research interests, one of which includes glycans and glycocalyx engineering. And I just want to ask you, what drew you specifically to this topic? And if you could elaborate on the importance of this topic.
That's a really great question. Sometimes I ask myself the same question, actually. Why am I doing what I'm doing? And I think it really comes down to what I find interesting. So if we back up to my undergrad, I was in a third-year chemistry course. I think it was called Organic Chemistry of Biological Compounds or something to that effect, taught by my eventual PhD thesis advisor, Professor Mark Nitz. And he was lecturing on carbohydrate chemistry. And it was the first time I had been exposed to how to manipulate the chemical structure of carbohydrates. What are the technologies we use to do that? And what might that enable? And for some reason, carbohydrates resonated with me. And I'm still to this day not exactly sure what it was that I found so shiny and attractive with sugars. But I generally like to focus on things that few others are deeply interested in. Not to say that there isn't a large carbohydrate community in the scientific realm. There absolutely is. But the roles of carbohydrates in the context of health and disease has, I feel comfortable saying, been quite a black box for most of human history. Carbohydrates are notoriously difficult to study. Importantly, they're not derived from a template, unlike nucleic acids and proteins, where you have a linear sequence that's decoded, maybe decoded again, and then you get a linear biopolymer that, say, folds into three-dimensional structures and maybe does some chemistry if it's an enzyme like a protein or a ribozyme. Carbohydrates don't behave like this. There's no template that dictates the structure of a complex carbohydrate, which is typically multiple monosaccharides linked together through covalent bonds. So we don't understand a lot of the details behind how specific glycan structures, carbohydrate structures, are even made, let alone what they do, or even where they are, or even their specific structures. We really now just have the technologies to start to deeply interrogate these things. Advances in mass spectrometry are illuminating the structures of carbohydrates that are clipped right off the surface of real human or mouse cells. We now have synthetic technologies, alluding back to what I started at initially, to not only make these structures, but also attach them in either natural or artificial linkages to other biomolecules like proteins or even the surface of cells. So I think we're entering a renaissance of productivity in biomedical research that's in part very much driven by a better understanding of glycobiology, which is really, I think, being driven by these new technologies and new tools that are emerging. And so because I have, I would say, rather interdisciplinary formal training, my lab's in a unique position to be able to combine traditional hardcore synthetic organic chemistry and then glycobiology, so immunology experiments, and combine those two disparate skill sets to do things that few others, at least in academia, can really do. And so, if you ask me what really wakes me up in the morning or has driven my curiosity for the later stages of my career, what I would say is just learning even a little bit about why cells spend so much energy producing these complex structures, linking them to function, and importantly, taking that information and using it to drive new therapies.
Yeah, pretty interesting. So in your article called Sialic Acid Ligands of CD28 Suppress Co-Stimulation of T Cells, you use T cell and dendritic cells matured with LPS to measure T cell activation by a specific antigen. I was wondering if you could explain how this model works and why it was the best choice for your study.
Great. Very good questions. I'd like to start by saying that while I am the first author on that paper, that was done under the mentorship of Professor James Paulson at Scripps Research. So I did that as a postdoc. So he's the senior author on that. To specifically address your question about T cells and dendritic cells and LPS, I think we need to have a brief conversation about how an antigen-specific immune response is mounted. And I think this is timely because, of course, everybody's thinking currently about how we mount immune responses, particularly against, say, viral antigens. And so learning about those fundamental mechanisms is integral to being able to explain the models we use and the technologies we use to explore those immune activation events. So for context, a T cell is a special type of white blood cell, a leukocyte, more specifically a lymphocyte. There are multiple what I'll call flavors of T cells, but for today's discussion, let's just keep it simple and say we're just concerned with a single type. T cells have a constellation of different proteins on their surface. And many of these proteins function to relay signals from the exterior of the cell, we call the extracellular space, to the interior of the cell, the intracellular space. So you can think of them as relaying information from the outside to the inside. Now, a lot of cells have these kinds of proteins on their surface, but T cells have some that are really quite special. In particular is one called the T cell receptor. It's not very creatively named, but that's what we call it, abbreviated the TCR. Now, this receptor is really interesting because your body actually produces a whole variety of T cells with different TCRs, many different versions of TCRs. And only some of those, what we call clones, if you will, or members of a polyclonal population of T cells, only some of them will actually recognize anything of biological significance. But through a complex process we won't get into today, once T cells are mature enough, they can enter circulation, and they eventually will meet what we call an antigen-presenting cell. You might think of these as classically being described as dendritic cells, or even B cells can be antigen-presenting cells. So let's link this randomized T cell receptor to an antigen-presenting cell now. Antigen-presenting cells display a very special type of protein on their surface called a major histocompatibility complex. In humans, it has a slightly different name. We call it human leukocyte antigen, or HLA. We call it MHC in pretty much every other organism, including mice, but they're degenerate for our purposes here. So an antigen-presenting cell with a major histocompatibility complex on it, an MHC, it will encounter biological material as part of its life cycle. Usually, we consider this to be proteins. So I'll give you an example. Say you have a bacterial infection. Okay? You've got an antigen-presenting cell floating around, and the bacteria have infected you. Some of them are dying and spilling their innards out into circulation. And the antigen-presenting cell can actually engulf some of that foreign biological material, internalize it, process it, and then load a short little fragment of that protein onto its MHC proteins, its major histocompatibility complexes, and then display that foreign material on its surface. So now you have an antigen-presenting cell that's floating around, and it's displaying this foreign biological material in the context of an MHC molecule on the surface of itself for all of these other immune cells in your body to see. And here's where the T cell comes in. So the T cell now floats around, and eventually it meets an antigen-presenting cell. Now, if the T cell just so happens to have what I'll call the quote-unquote correct T cell receptor, it will make a complex, what we call an immunological synapse, a cell-cell contact through the T cell receptor, ligating the major histocompatibility complex loaded with the foreign biological material. We call that foreign biological material an antigen. It could be coronavirus spike protein fragments. It could be from Vibrio cholera. It could be a tumor cell antigen. Unfortunately, it can also be healthy cell antigens, which is where autoimmunity comes from, but we'll get there in a moment. So now we've formed this immunological synapse. At this stage, if the T cell receptors formed a sufficiently high enough affinity interaction with that antigen MHC complex, a signal is relayed to the inside of the T cell through the inner part, what we call intracellular domain or cytosolic domain of the T cell receptor. Once that happens, a series of intracellular signaling events, which are quite complex and a little bit beyond the scope of our discussion, ultimately results in transcriptional changes inside the T cell, altering which proteins it's making, and ultimately resulting in a change of T cell phenotype, meaning how the T cell looks and behaves. But I'm skipping a few things here. First, it's not just a single T cell receptor, meaning a single MHC. There are an enormous number of these interactions that are occurring, duplicates of this. So many, maybe thousands of these interactions occurring at what we call the immunological synapse. In addition to that, the T cell receptor meeting its antigen MHC alone is not sufficient to fully activate the T cell, to fully stimulate it. There are other receptors at that interface that are essential for full T cell activation. In fact, if all you give the T cell is stimulation through its T cell receptor, ultimately the T cell becomes what we call anergic, and it actually becomes even more difficult to activate later. So it's not stimulatory. Immune system is full of these checks and balances, fine control. So we need other signals. Well, what are those other signals? This comes to the title of the paper. It's CD28 is one of these other proteins expressed on the surface of T cells. And it has a partner, what we call ligand, that's another protein that's tethered to the surface of the antigen presenting cell. So now when CD28 meets its ligand, which is CD80 or 86, expressed on the surface of the antigen presenting cell in concert with the TCR MHC complex, you now have what we call antigen specific signaling through TCR and co-stimulation through CD28. That's the 1-2 punch to tell the T cell, okay, this is a real signal, I've got to pay attention, and now I have to activate. I'm anthropomorphizing the system, but it's easier to talk about it this way. In addition to this, there's a third signal that's produced through soluble cytokines, which are signaling proteins that are usually exported into the extracellular space that will ligate other proteins on the surface of the T cell and antigen presenting cell. But to keep things simple, let's just talk about signals 1 and 2. So your question was specifically about, why we choose to use T cells with LPS matured dendritic cells to study this process. Well, we need to use dendritic cells that have been matured appropriately such that they are sufficiently able to present antigen on their surface in their MHCs, and also present enough of these ligands for CD28, so again, that's CD80 or 86, on their surface to be sufficiently stimulatory towards a T cell. This gets even more complicated because we use what I call primary cells for all of these experiments in the paper, where we're looking at the roles of carbohydrates in either increasing or decreasing the intensity of the T cell response. Primary cells are directly out of an organism, in this case, out of mice. But I told you earlier that an organism will have a whole complex repertoire of T cells with all these different T cell receptors. This semi-stochastic repertoire of antigen recognition capacity. That's not very helpful for doing basic science in a lot of situations, because we want to be able to give the system an antigen that we know what it is and we know how it will be presented. We can't just give ordinary mouse immune cells an arbitrary protein antigen and expect to get meaningful data out of that. So for these experiments, we actually used, in part, for the T cell side of things, animals that have been genetically engineered such that many of the T cells in their bodies actually have the same T cell receptor. It's a known T cell receptor that has a known specificity for, believe it or not, chicken egg white protein, ovalbumin. Why the heck would we use that? You might be wondering. Well, it's really a matter of being practical. Chicken egg white protein is really inexpensive. It's really easy to purify. And a lot of older immunology work has been done using that as a model antigen for those reasons. So we have these transgenic animals that we now have a whole bunch of T cells in them that we know what they recognize. They all recognize the same model antigen. So we can take dendritic cells out of another mouse or culture them from precursor cells from another mouse, give those dendritic cells chicken egg white protein, just let them sit in it for a while. They'll take it up, process it, display it on their MHCs, and then we can simply combine the T cells in those now ovalbumin or ova-loaded dendritic cells, and we, in very carefully controlled experiments, can measure if the T cells are more stimulated or less stimulated. For example, if we manipulate the carbohydrate content of the surface of the cells, which is what we did in that paper. And so ultimately, the major finding there was that we discovered that if you remove certain monosaccharides from terminal monosaccharides from some of these glycans, these sugars presented on the surface of either dendritic cells or the whole system, if you do that, you can actually manipulate the intensity of the T cell response, all other things being kept equal. Now, we weren't the first to show that, but we were the first to show that there may be a mechanistic explanation of this through co-stimulation. Because we have evidence in the paper that's presented that actually that second signaling event I described earlier, co-stimulation through CD28, meeting its ligand CD80 and 86, that event might be in part blocked by some of these glycan structures presented on the surface of the cells. And so when these two cell types come together, we know the protein-protein interactions are forming. And now we're starting to understand how this dense forest of carbohydrate material on the surface of cells might also contribute to tuning, fine-tuning these really critical signals at the cell-cell interface, the immunological synapse. And so I don't think it's too difficult to communicate the implications of this. If you can control the intensity of an antigen-specific immune response, you have a lot of therapeutic potential there. Because in settings where you want to augment an immune response, for example, against cancer, you can imagine removing some of these structures that are blocking stimulation. And in other cases, you might consider adding them to shut down an unwanted response. And that's exactly where my lab is focused right now. We're thinking about what I call glycoengineering strategies to now artificially manipulate these carbohydrates on the surface of cells that we now are starting to understand a bit of the functions and use that as a therapeutic handle to tune immune responses.
Thank you. That was a very well put answer because, one of the main reasons that we decided to do this podcast was to show students that, research papers are not as overwhelming as they might sound. So that's great to let our audience hear the logic behind the papers from the authors and the researchers themselves. So thank you so much for such a great explanation. And so speaking of T cells and antigens, in the same paper, in the method section, you mentioned the use of flow cytometry. And I know that us students, life sciences students at least, have heard of this assay, if I could call it that, at least once in one of our courses. So I just wanted to ask you if you could elaborate on how the process was performed, how long it took, if you could explain these things.
Absolutely. So I can speak to flow cytometry in a moment. I would really highlight again to actually your student listening population. Not only should we not be too scared of reading some of these maybe at first glance intimidating scientific articles, we should also be aware that this isn't done in isolation. This isn't done in a vacuum. I didn't do all of the experiments in that paper myself. I collaborated with experts whom were able to take a compartment of the study and really drive it to completion. So I made some major contributions to the study, which is why I was put as the first author on the paper, but it couldn't have been realized without help from some of my co-authors on that paper. So we should also be thinking about collaborative science and how by working with people that have complementary expertise to yourself, you can achieve things that would be impossible in isolation. So I'm really grateful to have had so many excellent collaborators throughout my career to help enable this really interdisciplinary science. So speaking specifically about flow cytometry, which is my major analytical technique as a scientist now, this is technology that's very simple to describe, but I think the data it gives you is a little bit abstract to process. So let me start at the beginning. So a flow cytometer, its mission is to give you information about a single individual cell from a complex mixture that could contain many, many millions of cells. So how does it do that? What we do is we take a mixture of cells, say I take a spleen out of an animal, and then I process it so that I have all the white blood cells from that spleen in a tube. I add what we call fluorescent antibodies into the tube with the cells. Now, an antibody, especially a monoclonal antibody, is a big protein, about 150 kilodaltons or more, depending on what we call the isotype. That protein will recognize some matter on a particular cell type. So many cells within our body, for example, T cells, are known to express certain proteins or other biological material on their surface. For example, the T cell receptor on a T cell, or CD80 and 86 on an antigen presenting cell. We have we have a lot of advantages now in 2022. We can buy a lot of antibodies from commercial sources that have excellent selectivity and specificity for a particular protein on a particular cell. So what we do is we buy these proteins conjugated, usually chemically conjugated, to a molecule that is light sensitive. We call them fluorophores. So for example, we have an antibody against, say, CD80. We know it will label antigen presenting cells. And it's covalently attached to a fluorophore that, when excited with a laser beam of a particular wavelength of light, will shoot out new photons of light at a lower energy, a longer wavelength. And those photons can be detected by some sort of technology incorporated into our flow cytometer. So how does this work? Well, we take the antibodies with fluorescent tags. We can take a whole panel of different antibodies against different proteins or other biomolecules. Each antibody has a unique fluorophore attached to it. We put them all in the tube with the cells. We do a little bit of processing, we wash them, and then we inject them onto our flow cytometer. And what happens is the flow cytometer has a very, very narrow fluidics channel on it. And I'm skipping a lot of steps here, but effectively what the machine is doing is pushing the cells into a sort of single file line along a very thin tube. And it's being pushed along in some kind of liquid, like a buffer, salt water or something. Now, individual cells will pass by an array of laser beams that are being shot across this tubing. When a cell passes across a particular laser that's being produced at a particular wavelength of light, many events can happen. One of those events is the laser beam will scatter. That scattering event tells us that a cell has entered the system. In addition, the laser will excite fluorophores that have the appropriate photophysical properties for that laser beam. And then we have a whole series of detectors that are at a different compartment of the instrument that are ready to accept the photons that are produced from each of those different antibodies in the system. So what we end up getting is a fluorescence readout of each individual cell within our mixture. Some cells will have a high fluorescence value for, say, CD80, which would then give us reason to think they're an antigen-presenting cell. And we're indirectly inferring expression by the fluorescence output. I know it sounds a bit indirect, but believe me, detecting specific matter on the surface of an individual cell is no small feat. So having these very specific antibody tools is really critical to realize this, even though they really are indirectly reporting on what we're interested in. So my lab, we own our own state-of-the-art spectral flow cytometer, which is a next generation version of this technology that allows us to look at far more parameters at a single time than you'd be able to on a traditional instrument. So we can profile what I call the immune landscape of animals that are either healthy or with disease. And we can pull out neutrophils, B cells, T cells, dendritic cells, eosinophils, pick a cell type you want. And then even more deeply profile each of those individual cells with respect to how activated are they? How antigen specific are they? Are they anergic? Have they been in a particular tissue compartment? Do they express a glycan we're interested in? And we can do all of that from a single tube. This instrument can collect 10,000, 20,000 events a second. So we get an enormous amount of information at the individual single cell level out of this instrument. Why single cells though? Well, I like to use an analogy to people's income for this. So say you enter a room and people make somewhere between $40,000 a year and $100,000 a year. And say the kind of median value of everybody's salary in the room is around, say, $70,000. Okay. Then Elon Musk enters the room, or Bill Gates enters the room. Okay, they have an annual salary in the billions of dollars. So now the median salary in the room I'm not sure what it would say. It's $100 million now. It's the median salary. Well, what does that number tell you? It tells you nothing, right? It's a totally arbitrary value because nobody in the room makes $100 million a year. The rich person makes $1 billion or more, and everybody else makes less than 100,000, say. Nobody makes that value. That's the difference between an average measurement and what we call a population level measurement. So if you could get information on every single person's salary within the room, you would then see that there are two peaks. There's a bunch of peaks clustered around 70,000 and a big peak, or a small peak rather, out at $1 billion. And that's the power of population level measurement. So the same rules apply to flow cytometry. Some cells will have high expression of proteins, other cells will have low expression. But if we took an average, we'd get a value that's actually not representative of any individual cell, very likely, depending on the system. So that's where the power of flow cytometry comes in, and other single cell analysis techniques like single cell sequencing and all of these new technologies. We can dissect complex systems at the level of the individual rather than relying on meaningless averages.
Yep. Thank you so much. So another question would be that one of the things in science and science findings, especially when it's like more clinical, is that there could be a lot of different interpretations of the results. So here's an interpretation, and is it accurate or not? So does the finding of your paper suggest that the more sialic acid there is in your body, the more suppressed your immune system is? And would that suggest that we should stop or like deprive our body from sialic acid?
Right. So just to give the listeners a little bit of context, the paper we discussed earlier with CD28 and T cell activation, the particular carbohydrate residue that we implicated in changing these immune responses is called sialic acid, which is probably not a carbohydrate most people have heard of before. It's rather unusual in the sense that it's actually 9 carbons instead of what we classically think of monosaccharides as being, which is 6. That's not always true, but for the purposes of today, we'll just say it's 6 normally. It's also charged, it's anionic, and it's usually found as the terminal monosaccharide on glycans in, say, the human body, okay? Sialic acid is actually a descriptor of a whole family of different 9-carbon monosaccharides that are anionic. So we're talking about a specific subset of that family, but we'll call them sialic acids for today. So by clipping those off, we were able to show that we get enhanced immune responses, as others have shown as well. So the question then is, if we have too much sialic acid in the body, do we have a suppressed immune system? And the answer to that, I would say, is broadly, it depends. So if you eat sialic acid, no, I sincerely doubt you're going to have immunosuppression. In fact, a lot of that probably won't even end up on glycan. Some of it might, if you eat it. Okay. The molecular and cellular context of that sialic acid is what's really going to drive its immunological function. So if you now all of a sudden, say, have antigen-presenting cells that have substantially more sialic acid on their surface, and that sialic acid's linked to particular glycan structures, because you have a whole array of structurally diverse glycans, carbohydrates on the surface of these immune cells, then yes, in that setting, there might be an immunosuppressive output. There's a whole rabbit hole of discussion we can go down there with other receptors that recognize sialic acid cap glycans. They're called the Siglex. Some of them are called the Siglex at least. And a lot of those receptors are indeed immunosuppressive. So if you have more carbohydrate ligands for those, you can actually decrease immune responses in the context of a cell-cell contact event, like an immunological synapse. But to say broadly, person X has more sialic acid than person Y, no, we need to know a lot more fine detail than that to start to generate a hypothesis about the immune activation state or potential of these different individuals. Does that answer your question? I think we covered it. Great.
Yeah, thank you so much. Again, I I just want to emphasize on how important it is for us that you as a researcher come and explain all of this to us and to our audience, because I know that it is overwhelming for students to contact researchers and, to read the papers and, understand their interests. So thank you so much. And I just want to ask you, as a researcher and a PI yourself, if you have any advice or tips for students who want to pursue research, not just in your field, maybe in any field in general.
Right. So that's always the magic question, right? Is there a formula for success? I'm afraid I don't have the universal solution or the universal comment there, but I can speak from my own experience, which I'll also remind everybody has been very linear, and I've been very privileged to be able to pursue the career path that I've pursued with really minimal barriers to success. That being said, it didn't just happen. There are a lot of parameters that have to coalesce for somebody to be fortunate enough to engage in really excellent science. The first parameter is the environment. You have to be in an environment you can succeed in. That certainly crosses between school and home life. But let's speak about school. If you can get some research experience in a lab that has a supportive mentor, that's a really great place to start. And I would be extremely fortunate throughout my entire career to have that. People will tell you to work hard. I agree that you've got to put the hours in. Research, I'm afraid to say, is extremely slow and often very frustrating. You need to put in the time to solve really boring technical problems. Let me give you some examples. So you think you need to express a protein. You're going to make a recombinant protein using the molecular biology techniques you learned in second year biochemistry. You go into a lab to do a summer research project, and the PI says, yeah, the plasmid's in the freezer. Just go ahead and express the protein. So you do it. You go, you transform your bacteria, you grow it up, you sonicate them. Then you go to purify the protein, and the protein isn't soluble. It precipitates out of solution. What do? You probably made the protein, but through no fault of your own and something that few could probably have predicted, you've got a bunch of white goo in the bottom of a tube. You can't do anything with that. It's not going to be active. Well, you just spent 4 months of your life getting to that point. So now you've got to think about, do I continue doing this? Do I stop? How can I solve the problem? Is that going to be fast or slow? Well, maybe you start mutating the protein. Well, now you've got to go back and make new plasmids and start all over again until you finally get, a year later, to a soluble version of that you need to do your actual experiment. That's the exciting part. I think the hallmark of a really good researcher is somebody who can make great scientific decisions, but can also deal with a lot of punishment. And I'm talking about research-focused punishment, meaning not everything's going to work. In fact, a lot more things are probably not going to work than are going to work, and certainly on the first try. So to be successful in this space, I would say you have to be okay with that. You have to acknowledge that that's going to happen. It's not a reflection on you as an individual or your academic performance per se. You make the best scientific decisions you can with the information available to you. You consult with knowledgeable colleagues, and you put the hours into the lab to eventually solve the problem. You will solve the problem. The result might not manifest the way you think it will. Projects change directions as you find out things are insoluble or soluble, stupid things like that. But stick-to-itiveness, durability, having that mental stamina, that mental fortitude, that's really important, because if you give up when things don't work, it's very hard to get anywhere meaningful in research.
Yep. Thank you so much. That's actually something that I guess undergrads don't really think about when they enter research. Not to discourage them, but something to consider.
Yeah, you definitely have to start somewhere. I encourage undergraduates who join my lab to try and stay for much longer than a summer research project or one semester. I I think very little can actually be accomplished in one semester, especially for somebody who has maybe never done any wet lab research before. So my recommendation to students maybe listening right now would be to try and find a position. It's hard, I know they're competitive, but if you can find a position where the PI is receptive to you staying for a more extended period of time, you might find it to be a more positive experience because you have a little bit more flexibility and, you know, maybe the results not manifesting in the one semester research project that you had officially arranged. So think about that for sure.
Yep. So one last question, and it's kind of like a custom at AMOSP in our podcast now, and that is, who is your favorite scientist and why?
Oh boy, that's a political question. Now I got to be careful what I say. Well, my favorite scientists are kind people. Fortunately, my mentors have been very kind individuals. I hope that I can be as kind as these people. I'm going to exclude my former mentors from this just so that I can be objective, I guess. A scientist that I take a lot of inspiration from is Professor Carolyn Bertozzi. She used to be at UC Berkeley. She's now at Stanford. Dr. Bertozzi has produced what are, in my opinion, some of the most critical advances in chemical biology. And she just so happens to do a lot of chemical glycobiology as well. So her accomplishments are extensive. She's won many, many awards very deservingly. I've met Dr. Bertozzi on numerous occasions. She's always been very kind, very open, willing to talk about science. She's a champion of equity and diversity in science, which I think more of us need to strive to recognize that there are major problems with that in academia and beyond. Dr. Bertozzi has produced research that spans disciplines. So for example, chemistry that allows us to make chemical bonds in water in the context of a complicated biological system, selectively make those bonds without inputting additional bond forming reagents. I know I'm speaking a little vaguely about that, but bioorthogonal chemistry, which albeit has had major contributions by a whole constellation of scientists, from the fundamental synthetic end. Actually, a lot of that work was done at my former postdoc institution, Scripps Research. But Dr. Bertozzi has really taken a lot of those ideas and turned them into a really useful tool for doing work in vivo, where you can't necessarily put a third input into a system and expect a bond to form. So that's really inspiring stuff. She's also been able to show that we can intercept glycan metabolism by providing mimics of particular carbohydrate building blocks. And those get processed by cells and displayed in the context of a complex glycan on the surface, providing unnatural chemical matter on the surface of a cell that we can do a whole lot of really interesting chemical biology with downstream. I'm not doing any of these major advances justice, but I really believe that she's one of the current scientific sphere's greatest scientists, especially in the chemical glycobiology space. So Dr. Bertozzi is my choice here.
Wow, that was a great answer. And on that note, I want to thank you again for accepting our invitation. Thank you again for coming. I wanted to see if you have any final words to share with our audience.
I just want to say that I'm so happy to see students like yourselves putting material like this together. I think that science has been opaque for a very long time. And I hope that these kinds of student-run events can help make it a little bit less intimidating. I'm really happy to discuss science in informal settings anytime. And I really hope that people learn something from today. And I'm always enjoy chatting with students. It's one of my favorite parts about being a professor now.